Welcome to Introduction to Microorganisms. This is Dr. Anup R. Markande. First, let us consider what are the characteristics of life. A life, when we say, it should show growth and development, reproduction and heredity. Heredity usually we say when there is genome, which is composed of DNA, which is packed in chromosomes, produces offspring sexually or asexually. It should also show metabolism, chemical or physical life processes, movement and or irritability, respond to internal or external stimuli, self-propulsion of many organisms, cell support, protection and storage mechanisms, which also includes cell walls, vacuoles, granules and other inclusions, transport of nutrients and waste. This is what we say when we are talking about characteristics of a living organism. So, characteristics of cells and life, when we say such things, Schwann and Sheldon were the first to say the cells as basic units of life. All living things, single and multicellular, are made up of cells that share some common characteristics, which includes basic shape, which is either spherical, cuboidal or cylindrical, internal content, which includes cytoplasm surrounded by a membrane, and a genetic material, mainly DNA in chromosome or chromosomes, also contains ribosomes, metabolic capabilities and others. So when we consider a cell is like an industry which is overall controlled by a central control panel, which is, we can call it as a nucleus. It is containing a blueprint of operations which is encrypted in coded message in DNA or as chromosomes. In this whole system, mitochondria give the energy. They are the energy source, and production machines are mainly endoplasmic reticulums, which produce either proteins, lipids, steroids or something like that. Production of requirements like carbohydrates and other energy sources are mainly by chloroplasts, and other arrangements or transportation of these produce is mainly by Golgi apparatus, while transport of these materials is by using vesicles. Boundary of work, the industrial walls is made up of, mainly in the cells, is made up of cell membrane, and other components also include lysosomes. So when we say an essential cell should have a wall that separates inside of the system from the environment. In this image, as you can see, it is a plasma membrane or a cell membrane. Organelles, as seen in this, is a eukaryote. So membranous organelles can be seen, while genetic blueprint, as said in before image, was in the form of crypted message, or a place for DNA is present in a nucleus, while ribosomes and other organelles smaller organelles are needed for protein synthesis and usual metabolism. So initially the classification was mainly named into taxonomy, the study of phylogenetic relationships between organisms, or the sorting of all organisms based on their relationship or differentiating features. We consider them in the form of kingdom, phylum, class, order, family, genus, and species. While genus is closely related species, while species are organisms sharing a set of biological traits and reproducing only with their exact kind. Further classification, especially with bacteria, is with respect to strain and a type. So when we say taxonomy, taxonomy relatively is easy to classify animals and plants based on their behavioral and appearance characteristics, which is an old school characterization on the basis of kingdom, phylum, class, order, family, genus, and species. When we, as you can see in this image, it is mainly for a squirrel. It is kingdom Animalia, phylum Chordata, class Mammalia, order Rodentia, family Squirrelidae, and genus and further species. Initially, the two major kingdoms were Plantae and Animalia. Animalia included all the animals, while Plantae included plant, mainly chloroplast containing organisms. The plant cell, just like any other cell as discussed before, has all the basic ingredients that are needed except an extra organelle, which is a chloroplast, and an enlarged vacuole, while animal cell has no extra organelles as seen with respect to plant cell. So then comes Anton van Leeuwenhoek. He was the first person to see microorganisms. Bacteria were first observed by Anton van Leeuwenhoek in 1676. The term bacteria, Greek for small stub, was first used in 1838. So there was around 150 years of difference. That difference is mainly filled by enthusiastic studying or enthusiastic seeing of these organisms by people who were not interested in actually studying these organisms but actually seeing them with curiosity, just like Anton van Leeuwenhoek. So, what are microbes? Microbes are invisible to the naked eye. You need a powerful microscope to see them, are everywhere around us, inside us, on us, in our food, in our homes, in the air we breathe, and the water we wash in, are mostly useful, but some are harmful, have been around for 3.8 billion years, and are vital for life on Earth. We will discuss about these points in our future endeavors. So, what do microorganisms look like? With respect to relative sizes and detection devices, when we consider microorganisms from humans to electron orbitals or atoms, microorganisms and usually a single cell, they fall exactly at the center. As you can see, a light microscope can easily distinguish a cell, a large eukaryotic cell, while the majority of microorganisms that we are going to study in future are made up of either bacteria, virus, or such small organisms which need very high resolution microscopes. For smaller molecules, we need an electron microscope to study them. So, what do microbes look like? When magnified hundred thousands of times and colored using dye, we see the bacteria, viruses, even one of the most deadly viruses. We can see these viruses in detail. Even the fungi and other organisms which are considered under microorganisms, we can see them. So microbes, they are good or bad? As you can see in the image, these microorganisms, since time immemorial means after Anton van Leeuwenhoek actually seen them, we consider them as bad, because whatever spoiled, microbes were always there. Whatever disease we saw, microbes were there. Whatever bad thing happened, microbes were there. So microbes were considered to be very bad things. Even in your ads, TV advertisements, you can see that many things which can kill microbes are easily sold. It's an advertisement strategy. But as you can see, many things in environment, from degradation of vegetables to diseases, are due to microorganisms and the stuff that they produce or the stuff that they help in degradation. But they are all not bad. There are microorganisms which are good and some which are extremely bad. So when we consider characteristics of cell, eukaryotic cells include animals, plants, fungi and protists. Contain double membrane bound nucleus with DNA chromosomes. These are with respect to eukaryotes. Contain membrane bound organelles that compartmentalize the cytoplasm and perform specific functions. Prokaryotic cells, which include bacteria and archaea, no nucleus or other membrane bound organelles. Mainly eukarya. Karya means nucleus, new. So true nucleus containing organisms are under eukarya, while prokarya, old or primitive nucleus containing organisms, are considered under prokaryotes. Initially, when Haeckel classified organisms after 1866, of course, microbes were already known after Anton van Leeuwenhoek's work in 1700. Haeckel's classification, as seen in this image with his own drawings, you can see it is mainly classified into Plantae, Protista and Animalia. Protista were classified with respect to the organisms which had microscopic structures, including diatoms, flagellates and other organisms which actually come under protozoa and diatoms. So after that comes 5 kingdom classification. 5 kingdom classification classified organisms into Monera, which are actually prokaryotic unicellular microorganisms, which mainly includes bacteria, archaea and other organisms, while protists mainly includes eukaryotic unicellular or multicellular organisms which are microscopic in nature, while the further classification actually includes plants, animals and fungi. Fungi, which fall between plants and microorganisms, are achlorophyllous and they are saprophytic or absorptive in nature, while animals and plant cells we have already seen, so they have their own characteristics which are distinct. So now we come to a new science that emerged after 5 kingdom classification, which is microbiology. What is a microbiology subject? It is nothing but study of microorganisms, organisms that exist as single cells or cell clusters which must be viewed individually with the aid of microorganisms, with the aid of microscope, are considered under microorganisms. You can see, as described here, individually, because we already see microorganisms every day as the teeth which we brush every day, the growth in between the teeth every day morning is the group of microorganisms that are growing together. It can be seen, it can be viewed without microscope. There is growth of microorganisms as microbial mat, as algal mat, as microbial film, fungi growing on bread. These are all which can be viewed non-individually but as a group, and hence the sentence here, definition here, merely says viewed individually with the aid of microscope. So, why study microbiology? Microbiology as a basic science. It is a basic science. It is in between zoology, botany, and other sciences which mainly deal with the living organisms. Microbiology deals with everything that is microscopic. Bacteria and yeast are useful in studying molecular biology, biochemistry, and genetics, while the important thing is that their ability to reproduce rapidly. They are genetically, that is DNA, and biochemically more simpler than higher organisms. They don't have much of these hassles while working with them. Working with bacteria and yeast for understanding the life processes has no ethical ramifications because they can be killed easily, they can be studied easily, they can be modified easily, and they can be manipulated easily. With respect to animals and with respect to higher organisms, it is difficult to understand the same ramifications, while we can actually study these in lower organisms and see them unfold in higher organisms in further studies. Microbiology as an applied science. Of course, like any scientific study, this has the ability to be used as applied course. When we already know that microorganisms have immense importance in medicine because they produce a lot of diseases and hence they themselves can be used as vaccines. So they are helpful in producing vaccines. They are also helpful in production of antibiotics, production of important biological chemicals like enzymes and insulins. In industries, they are helpful in production of many food beverages, wine, beer, and other things. Like even in milk industry and dairy industries, they are helpful in production of cheeses and yogurt and other things which we eat every day, which are giving nourishments to us now and then. Agriculture, maintenance of soil fertility, digestion in cattle, and all the things which are needed for usual working. Ecology or bioremediation. Microorganisms have been known for degrading food and other materials. Why can't we use them for degrading the things that are toxic, which are bad for the environment, which are bad for the ecosystem? These can be degraded by microorganisms, hence these can be used in bioremediation. So when we say microorganisms, it was not easy initially to classify microorganisms just based on their behavior and appearance as was done with respect to plants and animals. Advancements in DNA amplification and DNA sequencing has greatly helped in this regard. The phylogenetic relationship between microorganisms can be determined by sequencing the 16S and 18S ribosomal RNA of the organisms in question. Of course, 16S is with respect to prokaryotes and 18S with respect to eukaryotes. 16S and 18S ribosomal RNAs are extremely conserved and can be used for identifying microorganisms much more effectively. 18S ribosomal RNA is not just limited to microorganisms. This is also helpful in understanding higher organisms, including the higher vertebrates and higher mammals, because eukaryotic 18S ribosomal RNA is present in all the eukaryotes, even human beings. So we are coming to the three domains. All organisms belonging to one of the three domains depending on their characteristics. A domain is most inclusive, broadest taxonomic category. A single domain can contain one or more kingdoms, as you can see in this. Archaea, those are primitive microorganisms. Eubacteria, the true bacteria. Eukarya, which are true nucleus containing organisms. So these three domains of life are placed into domains and kingdoms based on their cell type, their ability to make food, and the number of cells in their bodies. For example, cell type, which includes prokaryotic or eukaryotic, while ability to make food mainly is in the form of heterotrophic or autotrophic mode of life. Number of cells in their body, unicellular or multicellular. So the three organisms can be further classified: prokaryotic and eukaryotic cells that lack nucleus or they have primitive nucleus, and cells that contain a definite nucleus. Unicellular, single celled, made up of one cell. Multicellular, made up of many cells. Autotrophic, can make their own food or energy needs, while heterotrophic, which cannot complete their energy needs with the machinery that they have in them. So this is what we consider as the three domain classification. The three domain classification is mainly classified into three major segments. First segment is eubacteria, second segment is archaebacteria, and third segment is eukarya. While all three are from a universal ancestor. So eubacteria and archaebacteria are prokaryotes, and eukarya is mainly eukaryotes. So eukaryotes, it includes everything which includes animals, plants, protozoa, fungi, everything. While archaebacteria, primitive organisms, as said before, and eubacteria, mainly bacteria. Archaea, these are very primitive forms of bacteria. Eubacteria, more advanced forms of bacteria, and eukarya, all life forms with eukaryotic cells. So microbes, when we say microbes, they exist in huge numbers. In one teaspoon, in single teaspoon of garden soil, there are over 100,000 microbes. In 1 liter of seawater, there are over 1 billion microbes. On your hands, there are more microbes than there are people in the world. There are so many microbes that scientists have only named 0.5% of them. Microbes outnumber all other species and make up the most of the living matter on the planet. So, as said before, the 16S and 18S studies are mainly with respect to some specific research-based studies like PCRs or polymerase chain reactions. These indicates up to 10,000 bacteria per gram of soil. Many bacteria have not been identified or characterized because they have not been cultured yet. They need special nutrients, are part of complex food chain requiring the products of their other bacteria. Need to be cultured to understand their metabolism and ecological role. So we are coming to domain Eukarya. So, domain Eukarya, as discussed before, they have definite organelles, plasma membrane, cell wall if present, made up of polysaccharides, like in presence of in fungi it is chitin, in plants or algae cellulose or pectins, in vertebrates usually there is no cell wall. Domain Eukarya can be further divided into fungi, protista, mainly single-cellular eukaryotes, plantae and animalia. When we consider eukaryotes versus prokaryotes have DNA in membrane nucleus, while prokaryotes have DNA in non-membrane nucleoid. Multiple chromosomes can be present in eukaryotes, while only one circular chromosome is present in a prokaryote. Eukaryotes have associated DNA with histones, while prokaryotes no histones, but histone-like proteins can be present. Eukaryotes organelles are present, while prokaryotes organelles may not be present. Mainly it has organelle, that is ribosome. Eukaryotes have cell wall, as discussed before. Prokaryotic cell wall mainly is made up of peptidoglycan. Eukaryotes have further cell division in the form of meiosis or mitosis. Prokaryotic division is asexual, either as binary fission, snapping, budding, etc. Eukaryotic motility is by flagellar whips, while motility in prokaryotes is mainly by flagellar spins. Now we come to domain Archaea. These are evolved from earliest life on Earth. These mainly include methanogens. These have the largest group, methanogens, and use CO2. They grow in extreme conditions, extreme environments. These are halophiles, which grow in high salt content, thermophiles, which grow in extreme temperature conditions, psychrophiles, which grow in extreme temperature conditions in lower temperatures, mainly freezing or below freezing, and acidophiles, which grow in acidic conditions, just as alkaliphiles, which grow in alkaline conditions. So the microbes, when we say microbes, there are major three groups, and these three groups are bacteria, viruses, and fungi. In this studies, or in this module, we are going to discuss introduction to microorganisms, mainly these three groups. So the course includes bacteriology, which mainly includes introduction, size and shape of these organisms, appendages, physiology of these organisms, reproduction, domain bacteria, and archaea. The mycology module includes introduction, reproduction and classification, major divisions within this group, interactions between the group and other organisms, mycotoxins, antifungal agents, and clinical mycology. In virology, we are going to study an outline of this group, replication strategies, methods within these studies, subviral particles, antiviral agents, bacterial viruses, and other viruses. So let us start with the next module, the first module, that is bacteriology.